our work focused on doing movement analysis more broadly using video. And so Gait is a part of that, but I just want to also let everybody know as we're going through the slides, if you have other behaviors or if you have other types of videos that you'd like to analyze, just stop me at any time and I'm happy to talk more about how we might be able to accommodate that. So happy to answer questions at the end, and I'm also happy to stop in the middle if people have comments or questions. But let me go ahead and get started. As Katie mentioned, I'm Ryan Remick. I'm an associate professor at the Kennedy Krieger Institute and at the Johns Hopkins University School of Medicine. On the Hopkins side, I'm in the Department of Physical Medicine and Rehab. My background, I'm not a clinician. My background is in engineering and movement science, and I've been here at Hopkins since about 2013, first as a postdoc and then transitioned to faculty. We have a three-dimensional motion capture lab that we use for kind of high quality, high resolution, granular movement assessment at the Kennedy Krieger Institute. But today I'm going to talk about some technologies that we've been using to essentially make that type of motion capture data collection available much more broadly. And so instead of having to come see us and undergo an hour-long session of putting all these little physical markers all over someone's body and tracking them as they move throughout the Gait lab or the motion analysis lab that we have, we're able to do that virtually now. So we're able to use some computer vision and artificial intelligence technologies to be able to do, oops, clicked the wrong button, to be able to do more virtual, we call them kind of motion capture in your pocket types of analyses. These are the types of technologies that I was just describing. What you can see here is a simple video of somebody just walking across the floor. Especially in conditions that are more difficult to recruit for participants in trials and more difficult to reach in general. We've been working with a lot of these technologies that can take a video like this and it can be recorded in virtually any setting and using any sort of video recording device. This is not a particular camera of any sort. This is just a camcorder you can see here set up in the corner of the room. And if we receive those videos, we can put them through some computer vision software that allow us to automatically track all these different anatomical landmarks that you can see here outlined by these little colorful dots. And so in my group, along with Jan Steinem, who's also on the call tonight, we've set up a lot of different pipelines to be able to take these little dots and hopefully extract something meaningful from them. So in our gait analysis pipelines, we can measure things like someone's walking speed, we can measure their step lengths, we can measure the kinematics of their joint movements. If you want to know how much somebody's knee, ankle, or hip flexes, we can do that too. And we've also done a lot of different testing to see basically when these technologies work and when they don't. So how these technologies work is that there was originally these large data sets of different images, and people manually went through and labeled all of these different anatomical landmarks in those images. And so that's useful from the perspective of there's a very kind of diverse, broad image set that was used to train these algorithms so that they're robust across a variety of different recording conditions. But one of the downsides is that there's not very much representation from patient data in those images. And so we've done a lot of testing to see if we have somebody walking like you can see here on the right. This gentleman is someone who's had a stroke. He has an ankle foot orthotic that he wears on his left leg here. And despite the presence of that bracing, we're still able to track his movement reasonably well. So even though this person appears fundamentally differently in this image than the images that were used to train the algorithm, we're still able to generalize and use it in these clinical populations. So that's been really helpful for us. Similarly, there are not a ton of images of kids in those training data sets. And so we've been involved in a variety of different pediatric focused studies at the Kennedy Krieger Institute. And so we've tested some little toddlers like you see here. And despite the fact that obviously her body is proportioned quite differently from an adult's body, we're able to track her successfully as well. As I mentioned earlier, we don't only do walking, so we also have some upper extremity models. This is me in my living room doing a quick repetitive hand open and close task that's common across several different neurologic exams. We've also done this testing in older adults with Parkinson's disease and older adults with stroke. I'm sorry, my PowerPoint crashed out. Let me bring it back up. But we've really started to look at a variety of different, we've validated this both in a variety of different clinical populations, and we've also validated it against our ground truth motion capture system. So a lot of the data that you're seeing here with these videos, we'll have simultaneous ground truth motion capture data that we can use to assess how well the videos are able to do the movement tracking. And so often people will ask, do we need the latest and greatest recording devices to be able to do this? One thing we've also done is look at kind of the other complete end of the spectrum and use some of the oldest videos we could find. So this is actually one of the oldest known recordings of somebody with Parkinson's disease. This video is over 100 years old. And you can see that despite the poor video quality and the black and white nature of the video, we're still able to track the tremulous movements that you see in the patient here. So again, really any video recording device, as long as we can get a good view of the patient or research participant that we're trying to track, we can use a lot of different sorts of video sources. We have been really focused on this. As I mentioned, I'm in the rehab department here at Hopkins, and so we have really been focused on using this in a way that could really facilitate improved telerehabilitation and kinematic motor assessments remotely. And so we are not, I always put it up front, we're not really trying to diagnose anything or make decisions for the clinician. Rather, all of our work has been set up so far to really try to give the clinician or the researcher better data that they can use to inform their clinical decision making or their research study. And so I always show this image that we've made with the clinician kind of in the center of the whole workflow. We still want the clinician and the researcher to be at the center of this. And again, the goal is to provide better data that we can then use to improve our studies and improve clinical care. As going through this work, we've also done a ton of feedback gathering to try to figure out what kind of technologies people actually want to use. And so here are the design constraints we've been working on. Importantly, these technologies that we're putting together don't have any additional requirements as far as new equipment or setup. We try to minimize the amount of time that needs to be spent beyond a current clinical standard for doing these types of recordings. They're all possible on a single accessible device. People just continue to tell us they basically want to pull their device out of their pocket and be able to use that without having to, again, start to use any new equipment that's kind of outside of their current workflow. And then these last two we're really still trying to optimize things to provide easily interpretable results in this ultimate kind of point and click solution where, again, you could pull your phone out of your pocket, take a video of your patient or research participant, and get something useful back. I've talked about this a little bit already, but here's some commonly asked questions of how accurate it is that we've done some validation work to establish that. Is it validated in my patient group of interest? We have done validation in both pediatric and adult populations. Talking about that at the top, can it measure my behavior of interest? We have probably the most data on walking, but as I also showed earlier, a wide variety of different upper limb videos too. So if there's anything that's of particular interest to this group, I hope you'll please let me know. And then usability is the big thing. Like, how do we actually use this? And so that's where we spend a lot of our time, is trying to make this as easy as possible to use. So again, if we take these recordings where we have the video on the top and we have the motion capture data on the bottom, we can make these direct comparisons to do the validation. And then here's just some quick, I know we're a little short on time. We have a bunch of data like this where you can see the motion capture versus the video-based data. We see strong correlations and strong agreement across nearly every gait parameter that we've been studying in our work so far anyway. This is in adults without motor impairment, but we've done the same thing in a couple of clinical populations as well. These are more step lengths. So again, the previous slide was step time. This is step length, still good agreement there. And then also walking speed. We see tight correlations between the motion capture and the video-based data. Quickly, here's those sagittal kinematics. So if you're interested in joint angles, like the sagittal hip angle or sagittal knee angle, sagittal ankle angle, we also see pretty close agreement between the video-based and the ground truth motion capture approaches. And as I mentioned, focusing on usability, we have all of this software currently freely available on the internet. So anybody can hop on and download it and start to use it. And of course, we're more than happy to set up some tutorials or training sessions, whatever might be most useful for this group once people start to analyze these videos. I think for the sake of time, I know we want to have a little bit of Q&A, so I will stop here and just be happy to answer any questions that people may have.
Thank you so much. A couple of comments to open up the discussion. I want to sort of bring back to this protocol, and pardon my darkening light and background here. I'm not in the usual place where I'm in my house. But what I wanted to raise was that we know that motor function, movement, walking ability is something that we hear is really important to patients and families. We know that the one drug that the indication is for prevention of loss of ambulation. So honing in on that as a critical functional measure some of the goal was to make sure that we have ways to deploy highly quantitative approaches to assessing gait within this natural history study, and then also to use strategies that everyone can use everywhere, and that maybe even could be independently assessed at a future time by another group. There was in the data, the objectives, the calculations were sort of the same everywhere as compared to some of the things that are quite variable, like 6 or 10-meter walk, things that have a lot of user variability. This removes some of that. The second thing that I want to mention is that we know that some of you do have pressure sensitive gait mats and other kinds of equipment, but not everyone does. So as we go to think about what's available at each site, we think of this video capture as a core way that everyone can be collecting the same kind of data. We'll ask you about some of your room or hallway dimensions so that we can think about what the setup might look like. And then we'll also be asking if you have any other kinds of equipment that we should be aware of. And so we can think about whether or not there's some cross-validation opportunities at certain sites within the study. So with that, why don't we open up for questions that people have? And thank you again for this introduction. It's not the last time that we'll talk about this topic, but it's a nice kind of beginning to thinking about making sure we all understand the methods and the strategies that we'll use. Questions?
I've got a question, Ryan. G'day, I'm Adam from Melbourne. What level of fidelity do you have in terms of things like finger movement? Yeah, I think you can see where I'm going with
Yeah,
that.
pretty good. Pretty good. We've done a lot of finger tapping. We've done, as I showed earlier, the hand opening and close. We're always a little bit at the mercy of the sampling rate of the video, but for a standard iPhone, 30 hertz video, we can usually get estimates of tapping frequency or something like that within, oh, I don't even, essentially within the sampling rate of that video. And so we've done quite a bit of, because we work a lot with stroke, we've also done some finger individuation stuff where if you're trying to have people isolate the movements of each finger, how much the other fingers move along with that. And we've also seen pretty high granularity in those results too. So yeah, if you have any ideas, ask away. We're happy to help. There was a comment in the chat, yeah, about the patient confidentiality. And so that is where we meet a lot with the IRB to essentially what we have done this purely for research purposes. So everybody has consented to being recorded in these videos and having the videos analyzed. Our videos live on Hopkins OneDrive, which is the kind of Microsoft supported cloud storage. And so that's how we have handled it. We have not if it is necessary, we haven't played with these, but there are some new AI technologies that'll automatically blur the faces in the videos. And so if you just need to remove the identities that way, it is possible. We've looked around at it a little bit, but haven't used it yet.
Yeah, I think that will be an issue. Sharing data, like all our families will consent. Oh yeah, you can walk back and forth. It's sending the data and making sure that they're okay with us sending the data. And how do you store the data? I'm sure I already know what my IRB is gonna say, and then who's gonna see the data, right? So
yeah,
I think those are things that whoever's doing the consent right now needs to know, because some centers are a little bit more picky than others, but I could just see that now, like
yeah.
especially international data, is another international team going to have access to that? So yeah, and I'm sure you guys are thinking about that already as we speak.
Yeah, we've done several interinstitutional collaborations on this type of stuff. And so usually one of the easiest ways to get at that is to avoid sharing the videos at all if possible. So that means oftentimes training different sites to implement the software. And usually it takes us 10 to 15 minutes to get someone up and running if they're able to download some of the software in advance. And so that's one possibility, is that the data that could be shared would be the de-identified outcomes, the gate parameters and that kind of thing, but not the actual raw video themselves. I don't know if there's interest among this group in kind of like amassing a large video data set. But if not, and everybody is okay with kind of having a more distributed approach to the video collection, it would be possible for us to have a training session or something for everyone, or a tutorial to get people up and running with it. But I'll defer to those who are making the decisions about how the data are all getting put together.
Yeah, we definitely welcome input on the consent. I know that the consent work group is thinking about this, and there have been some requests for volunteers to work on that consent work group. Even if you are not looking to join another work group, we do ask that if you have templates, information, suggestions as it relates to consent content, please reach out, because we are thinking about those things now. In terms of sharing, there are an overarching sort of data sharing structure within the consortium and with the DMCC that is part of the study overall. There's also a long-term data sharing requirements. So this is an NIH-funded study. So there are long-term data sharing and a data management plan. In that plan, just as Ryan said, there's no plan to share the actual raw video, but rather to share either derived variables or other non-identifiable pieces. But within the consortium, we do need to be able to share video so that we can think about our assessments and fidelity assessment, or again, the actual data that we might be collecting. So there are a lot of different kinds of recordings that are being taken, whether they are video or audio recordings. Even audio recordings are theoretically it's voice, it's an identifier. So the consent, the goal is for that clear, that what kinds of sharing might happen, and then also to allow people to opt out. So if they're not comfortable having video taken or an audio recording taken, they still are able to actively opt in or opt out of that particular activity and still participate in the rest of the study. We recognize not everyone is going to be comfortable with that level of sharing. We hope that some will. And I think I see a discussion in the chat that we'll want to definitely return to make sure that we are thinking about some of these other computational questions as well, both data and transfer in transit and then storage. Mariana, it looks like you have a question. There's a question about so many of our participants may have equipment like a vision cane, or may have one person assist either for motor function or as a vision guide. And so what does that look like in terms of open pose and computer vision as you've discussed it?
Yeah, it's usually not a problem if in those specific, and the times that we really start to have issues are sometimes with a walker. But if you have a cane or some kind of more kind of a single isolated type of device, that usually is not a big issue. If there are more than one, if there's multiple people in the frame, Jan has actually put together a workflow to be able to isolate the person that you want to be analyzing and track that over time too. So both of those things are usually not issues. In a lot of the patient populations that we have worked with there, those are common occurrences as well. So happy to help others troubleshoot if you have some of those situations.
Well, thank you very much. If there are additional questions, again, please continue to put them into the chat. There are some that we'll try to do a Q&A and get that back to everyone with whether it's about our biomarker or biospecimen collection discussion or about this introductory discussion. We'll certainly follow up with any questions that are unanswered.